Hello dear student, this is Mohit Yadav and today we will talk about biochemistry. This is 11th class which is very important for pre-board as well as competitive exams. So guys, we can say the biomolecules come under the biochemistry branch. Biochemistry is the branch of science which deals with the study of biomolecules. So, student becomes on biomolecules. Biomolecules. Students, as you know, biomolecules is very important for all living organisms. As you know that first of all molecules form, and after that cell organelles, and after that cell and after that tissue, and after that tissue system, and after that organ, and organ form organ system, and and student organ system form body of livings. So student, this is the format to form a body. First of molecules, and molecules form cell organelles, and cell organelles form cell, and cell form tissue, and tissue form tissue system, and tissue system form organ, and organ form organ system, and organ system form body. So student, we will talk about a cell. This is a cell, and in cell organic and inorganic both are found. Organic and inorganic substances are found, and student both are called cellular pool. So students, as you know, in a cell organic and inorganic matter are present, and both are called cellular pool. Only organic matter is called biomolecules. Only organic matter. Student, we are going to talk about types of biomolecules. Types of biomolecules. Types of biomolecules. Students, on the basis of molecular weight, biomolecules are two types, such as microbiomolecules and macrobiomolecules. Microbiomolecules and macrobiomolecules. Student, this is the on the basis of molecular weight. Student, biomolecules are those components which molecular weight is approx 18 Dalton to 800 Dalton. Its molecular weight molecular weight is approx 18 Dalton to 800 Dalton, and macromolecules is molecular weight is approx 10,000 Dalton. 10,000 Dalton. So, students, on the basis of molecular weight, there are two types of biomolecules: microbiomolecules and macrobiomolecules. Microbiomolecules are those components which molecular weight is approx 18 Dalton to 800 Daltons, and macrobiomolecules is molecular weight is approx 10,000 Dalton. So, student, we can say example, that is amino acids, glycerol, and fatty acids, and nucleotides and nucleosides and monosaccharides. This is the example of microbiomolecules, student. If and macrobiomolecules is the polymer of microbiomolecules. So, student, polymer of amino acid is called proteins. So, we can say proteins is the macrobiomolecules, and glycerol and fatty acid is the polymer of lipids. Student, lipids is the exception of both microbiomolecules and macrobiomolecules, and nucleotides and nucleoside polymers is called nucleic acid, such as DNA. Students, this is the monosaccharides, and polymer of monosaccharides form carbohydrates. So, this is the student example of microbiomolecules, and this is the example of macrobiomolecules. Microbiomolecules is those biomolecules which molecular weight is approx 18 Dalton to 800 Dalton, and macrobiomolecules is molecular weight. So, guys, this is the example of microbiomolecules: amino acid, glycerol, fatty acid, nucleotides, nucleosides, and monosaccharides. And the polymer of these substances is formed macrobiomolecules. Polymers means is the formation of long chain. Such as many amino acids polymer form proteins, and glycerol and fatty acids polymers form lipids, and nucleotides and nucleosides polymer is formed nucleic acid. In nucleic acid, DNA and RNA be studied, and monosaccharides polymer form carbohydrates. But students, lipids is the exception between both macrobiomolecules and microbiomolecules. We are known that what is biomolecules, and this is a question: how can you separate biomolecules and how you can separate macrobiomolecules and microbiomolecules from a living cell. Student, let's separate biomolecules, that means macrobiomolecules and microbiomolecules from a living cell. This is the question that can how can you separate macrobiomolecules and microbiomolecules from a living cell, from a living cell. Question first. This is question. Firstly, we have to take a living cell. A living cell. Then this cell should be crushed. This cell should be crushed with the help of mortar and pestle. With the help of mortar and pestle. And after crushing the living cells, we get slurry. We get slurry. And in this slurry, we have to add dilute solvent. Dilute solvent. Dilute solvent. After that, we make a setup. We make a setup. This is the student setup. And in this setup, this slurry drop. And in this setup, this slurry is dropped. This setup is work on the basis of filtrate or molecular weight. This setup is work on the basis of molecular weight. So student, this slurry is dropped in this setup, and those molecules filter, and those molecules filter is called filtrate. Is called filtrate, and it is also called soluble solvent. Soluble solvent. Acidic soluble solvent. Acidic soluble solvent, and this is called microbiomolecules. Microbiomolecules. This is called microbiomolecules because the molecules filtrate. So it is also called filtrate or acidic soluble solvent. And student, those molecules which are not filtrate from this setup is called acidic non-solvent acidic non-solvent, or it is also called retentant. 10, and this molecules is called macromolecules. Macrobiomolecules. Macrobiomolecules. So students, this question is can be proved that firstly we have to take a living cell, and this living cell is crushed with the help of mortar and pestle, and after that we get slurry, and in this slurry we have to add dilute solvent. Dilute solvent means DDTA, and after that we make a setup, and in this setup this slurry is dropped, and those molecules which filtrate from this setup is called acidic soluble solvent or filtrate, because it is filtrate. This is called microbiomolecules. And students, those molecules which not filtrate from this setup is called retentant or non-solvent or non